I'm going to be talking a little bit about combining neuropixels with wide field imaging. So this is a little bit of a niche subject because it requires you to have a lot of things combined at once. It's not a standard kind of recording setup at the moment. But it allows you to do some pretty interesting science and answer some questions that you wouldn't be able to otherwise. So even if you're not going to do this in your own lab, I'll show you an example of what I do and what kinds of questions I use it to answer. So in my setup, I do Neuropixels with wide field imaging, and I do that by having a prep where I remove the top of the mouse's scalp and I expose the entire skull. On top of that, I put a head plate holder, a head plate in the back, which is kind of the standard setup. And then I have this plastic well around the skull like so. So now I have this open skull in the middle here with this head plate in the back and this plastic bit in the front. Now I'll fill this area where the skull is with a little bit of glue. And then on top of that, I'll put this UV glue. And so what that allows it to do is gives it this really hard protective coating that you can see through with the light. And then when I go to do my Neuropixels recordings, I drill a craniotomy in the skull, and then I can fill this whole well up with saline or ACSF when I do my recordings. So this is the general outline of what's going on the mouse that allows me to do the wide field and the Neuropixels at the same time. So when I actually do my recording, I come in from the side with this Neuropixels probe, and then on the top, I've got my wide field imaging. So the main kind of restrictions I have in this setup are having the Neuropixels probe at a large enough angle that it kind of stays out of the way of both the mouse and the microscope. So I have a kind of narrower window than one might normally have to record. All right, one thing I want to touch on is the idea of light artifacts on Neuropixels probes. So I think that this is something which has been mentioned previously in the course, but Neuropixels probes are super sensitive to light, I guess, because it's the same circuit as in a CCD camera, which is why they're called Neuropixels in the first place. So when you shine a light on these things, you're going to have a huge artifact. In my case, I'm using these two lights. I use blue lights to get at GCamp calcium sensitive activity and violet light to get at GCamp calcium insensitive activity. So this essentially gives me a measure of the blood flow, and this gives me a measure of the activity. And I use those to get rid of the blood artifacts. So if you were delivering this light normally, it would just come on as a pulse like this. In my case, that lasts about 6 milliseconds. If you want to reduce light artifacts on the probe, the easiest way to do it is to ramp the light. So in this case, I'm starting at 0 and I'm quickly ramping this up over the course of 1 millisecond. So it's a quick ramp, but it takes a little bit of time to reach the top. So adding this ramp here is a good way to reduce the light artifact on the probe. To give you an idea of what these light artifacts look like, what I'm showing you here is a raw trace of the LFP of one of the channels on the Neuropixels probe. So you have these huge kind of deflections, these square deflections that go up and down. So every time the light from the wide field camera comes on, you'll get a deflection on the Neuropixels probe. So if you take the average LFP around the light onset and offset, you get something that looks like this. It's essentially a step function where you have the probe have this downward deflection with this little peak here, and then it comes to a steady state. When light goes off, you have the same kind of thing. And you get a different amount depending on which color light it is. All right, so one thing we can do is we can just take the average response to the light or the median response to the light, and we can subtract it every time we know that there's a light pulse, and you do a pretty good job of getting rid of this activity in the LFP. So when I clean the LFP activity, that's one of the ways you can do it. You just get the average light response, you subtract it, and then you have a relatively clean signal. In the action potential band, you can see a similar thing where this now is a longer time course, and you can see that there's just these crazy peaks all over the place. So these are all light artifacts. We have no idea where the spikes are at this point. So if we want to try to clean out these light artifacts, one way to do it is just with local median subtraction. So in other words, I'll take a bunch of channels around this channel. So, maybe 50, maybe 100. I'll take the median response across all of those channels and I'll just subtract it off. After I do that, I'm left with something which is pretty clean. So you can see the light artifacts are substantially reduced. But these little peaks here still are not usually spikes. These are the little on and offsets of the light. So we can further clean that up if we then just zero out the times at the light onset and offset. And once we do this, then we can finally get down to spikes. Okay, so the thing I want to show essentially with this slide is that the Neuropixels probes are very sensitive to light. You get these huge light artifacts, but you can usually do a pretty good job of cleaning them up either by median subtraction or some other kind of thing like that. You can have this trace, which looks like it's totally full of light artifacts, and get down to neurons at the end of the day. So it kind of convinces us that the information is there to do both imaging and electrophysiology at the same time, even though our raw electrophysiology taste looks insane. So the most common way I get rid of these artifacts is, as I said in the last slide, just with common average subtraction. So I'll show you, for example, here, the raw values across a number of channels. This is the action potential band. Over here in this white zone here, this is before the light comes on. And then as soon as the light starts to come on, you'll see these flickerings of blue and red that essentially dominate all of the recording. So if all I do then is I take the median across these channels and I subtract it from every channel, I can do a pretty good job of getting back a signal which looks reasonable. So that's shown down here. And now you can see some kind of more diverse red and blue that looks kind of on par before and after the light starts to come on. So when I clean up my data, this is actually all I do. I just do common median subtraction. And then that gives me a pretty clean trace. With that trace, I put it into Kilasort, and then Kilasort is going to pick out things like this, which are artifacts at the onset and offset of light, and then I'll be able to throw those away as clearly noise. So an example of that is shown here. When I run this common average subtracted trace through Kilasort, and then I look at the result in phi, some of my units look like this. They're clearly not units. They're these crazy things, and they also correlate with when the light came on and off. So I know that these are very obviously artifacts, and I just throw them out. So what that means is that I can do a pretty good job of going from this crazy, noisy signal to getting pretty good spikes, despite the Neuropixels probes being so sensitive to light. All right, so the types of experiments that we can do then are putting the probe in a couple different places and then looking at how that relates to activity across the cortex using this wide field calcium imaging. So the first example I'll show you is just putting the probe in the cortex and looking at the relationship between multi-unit activity in the cortex and fluorescence activity in the cortex. So what this gives us is an ability to essentially calibrate what the GCamp signal is telling us about the spikes within the cortex. So this is one kind of scientific question that we can get at by combining these two technologies. So the nice thing about the Neuropixels probes is that they're so small that they're essentially invisible in the imaging. So in this image here, the craniotomy is here. It's almost impossible to see. And then the probe is right here, this very thin line. So I'm going to outline these two things. The craniotomy is this little circle, and the probe is almost impossible to see. It's this tiny little line. So that's one of the wonderful things about these probes is that they're so small that you can have them in the brain at the same time as imaging and essentially not obstruct the brain at all. All right, so when we put the probe in the cortex so that we can get both spikes and calcium imaging in the cortex at the same time, we can look at the relationship between these two signals. So this was in a paper that came out last year, and the idea was just to show that there's a strong correlation between fluorescence and spikes. So in this case, we could take a fluorescence trace within our ROI here, and then we can take the multi-unit trace that we're getting along the probe, and we can look at how correlated their signals are across a different range of frequencies. So in this case, we're doing that both with the multi-unit and the LFP. And essentially what this is telling us is that up to 10 hertz, the fluorescence trace is able to very nicely follow the multi-unit trace. There's a lot of coherence within this 10 hertz, 0 to 10 hertz band. So essentially everything that's faster than 10 hertz, the GCamp isn't able to keep up with, but anything that's slower than that, we have a really nice correlation between fluorescence and multi-unit. So in this particular paper, Alina was the first author, and she was focusing on 3 to 6 hertz oscillations in the calcium signal. And we use this to show that the 3 to 6 hertz oscillations that you see in the fluorescence are in fact correlated to 3 to 6 hertz oscillations in spikes. So the fluorescence is a nice readout in that case of spikes. Another thing that we can do with this is to try to come up with a deconvolution kernel to go from fluorescence to spikes. So one of the things that you might have noticed is that this fluorescence signal doesn't really look a whole lot like this multi-unit signal. And that's because it's convolved with this GCamp decay. In other words, when there's a spike happens, the GCamp goes up and then it slowly comes down. So this is just a kind of slowed down version of this multi-unit activity. So we can come up with a kernel then to essentially deconvolve the fluorescence to get something which looks more like the multi-unit activity. So if we just do simple linear regression to try to come up with the best combination of fluorescence time courses which can recapitulate this multi-unit activity, we come up with a kernel that looks like this. So by taking this kernel and moving it across the whole fluorescence trace, we can then get what's essentially a deconvolved fluorescence. And now this deconvolved fluorescence looks much more like our multi-unit signal. So the thing that this dual imaging and electrophysiology has brought us in the cortex is A, it shows us that the fluorescence and the multi-unit signals generally are good reflections of one another. And it also lets us go from raw fluorescence to this deconvolved fluorescence that gives us a much more multi-unit looking signal. So in that experiment, we were putting our probe in the cortex and we were also imaging in the cortex. My question was to put the probe deeper in the brain and look at the relationship between cortical activity and subcortical structures. So in my case, this is specifically with the striatum. The idea is that you have all of these nice parcellated areas in the cortex, in the striatum. There's kind of a question as to how you should parse these areas or how they should be related to striatal activity. One of the things we do know is that you should have this part of the striatum, which is related to the visual cortex, and then you should have potentially different signals between the cortex and the striatum in these two regions. So in other words, maybe when the animal is paying attention to a stimulus, you get a large response in the striatum, but it doesn't when the animal is not paying attention to the stimulus. So the idea was that you have this visual area in the cortex, this visual area in the striatum. We wanted to see how the activity between these two things was related. On the other hand, you also have this motor region in the cortex, which should be correlated to this motor region in the striatum. And the idea, again, was to look at relationships between the activity in these two structures. So the hypothesis here was that the cortex might be active throughout the duration of a movement, whereas maybe the striatum exhibits some bracketing activity. Okay, so this is all just a general overview to say that by combining wide field and electrophysiology in this particular case, we can get the activity of the cortex simultaneously with the activity in the striatum using neuropixels, and we can get the relationship in activity from one to the other. All right, so when I do this experiment, it's very similar to the setup I showed you before with the cortical recording. The only difference is now, instead of putting my probe in the cortex, I'm going to put it in the striatum. So I'm coming again from the side and I'm putting my probe at this 45-degree angle. And the idea is to hit both the visual and the motor aspects of the striatum. While I do that, I also have this wide field imaging so that I can get the activity of the whole dorsal surface of the cortex. All right, so one of the things that is nice about what this buys you is the ability to align electrophysiology. So we've talked a little bit today about how to go from, for example, your histology to where you are in the CCF space. And one of the things I think that has come across, especially like when Mayo was talking about how to align these two things, is that it's a little bit tricky to figure out exactly where your probe is relative to the brain, because maybe it's not exactly where your histology looked like, or maybe it was a little bit deeper, a little bit farther up. So when you're looking at your raw electrophysiology data, it's essentially just this whole bunch of spikes that, in my case, I think that this is in the striatum because I have some electrophysiological landmarks above and below. But other than that, I don't really have a super clean idea of where I am. So in other words, I want to be able to take these spikes up here and say precisely where am I within the striatum. So historically, kind of what people have done is they've put their probe in the medial part of the striatum. In other words, maybe they're aiming for a place like this, but as Anne said, sometimes you miss. In fact, often you miss, and your probe goes into places that you don't necessarily intend to go. So we want to be able to have a robust way to go from these spikes to a precise location, in this case within the striatum. And this gives us the general power to say, what do I combine across sessions? So in my case, for example, I want to do the same recording, let's say 50 times. And in each of those 50 recordings, I want to be able to say, when was this part of the probe in the dorsomedial striatum? When was it in the dorsolateral striatum? And then how do I combine those across recordings to average and give myself the ability to do some nice statistical analysis? So as I said before, sometimes what people have aimed for is the dorsomedial striatum. I put in quotes here because that could end up in a lot of different places. But we want to avoid something like this and instead go for a much cleaner distinction of regions. So we want to say region 1 is wherever my probe was exactly in this particular region, and then region 2 and region 3. Sometimes my probe will be here, sometimes my probe will be here. It'll really have a lot of variability depending on the day, but we want to be able to do this relatively robustly. So the way that I do that is by, similar to coming up with the deconvolution kernel I showed you before, we want to move from wide field fluorescence to spikes in the striatum. So in other words, let's say I have my fluorescence across the cortex down here, and up here, I'm taking all of the spikes that live in this dorsomedial part of the striatum. So I can give you a schematic of that down here. My wide field data will look something like this, and then my multi-unit activity in the striatum will look something like this. All right, now, how can I best get from these images down here to this multi-unit activity up here? We can figure that out, again, using linear regression to come up with a spatiotemporal kernel that just transforms these images into this line trace. In that case, when we take this kernel and we move it across all these images, we'll be able to recapitulate this multi-unit activity. All right, so that gives us a kernel on the cortex in space and time that best predicts activity within this part of the striatum. So I'll play that as a movie now. So what you're seeing up here is time before the spike, time after the spike, it's generally blank, but then right essentially at a lag of 0, you have this nice relationship where certain parts of the cortex are very predictive of this part of the striatum. So then if I show you a still frame, we can get this map. And what this map is showing you is that in this particular recording, when I pool parts of the probe that live in this part of the striatum, it gives me a map of the cortex that looks something like this. All right, so now we can take this map as essentially a precise marker of where our probe was within the striatum. What I mean by that is, if we take different parts of the striatum, here color-coded in this rainbow gradient, and we look at their associated cortical map, we have very specific topography. Whereas the dorsomedial striatum has, for example, secondary motor regions, the dorsolateral striatum has these orofacial motor regions. This also ends up giving us a cleaner signal than if we did spike-triggered averaging, but they're both kind of the same general principle. So at the end of the day, what we get then is this gradient from dorsomedial to dorsolateral striatum, which is associated with this gradient from these sensory to these motor regions in the cortex. All right, now what this allows us to do is to use these maps to figure out precisely where our probe was during our recording. So coming back to this question we asked before of what am I combining across sessions, we can say, if we take a particular segment of our probe and the map looks like this, now we'll be able to call it dorsomedial striatum. Likewise, we can have one for the dorsocentral striatum, and we can have one for the dorsolateral striatum. So in other words, sometimes my probe will be over here, and sometimes it'll be over here. But if the map looks like this in the cortex, then that's a pretty robust marker of the fact that I was in what I'm calling, in this case, dorsomedial striatum. So we can now answer this question of where am I? If I make a map in the cortex of all of these different places along the probe, I can say that this part of the striatum was associated with this map. So I'm going to call that dorsomedial striatum. whereas these spikes were associated with this map, dorsicentral striatum, and these guys were associated with this map, dorsolateral striatum. So I'm going to have different placements of these along the probe and in different amounts every time I do a recording, because every time you do a recording, you're going to be in a slightly different position. But what this allows you to do by combining imaging with electrophysiology is have a robust relationship between where I am in this particular recording and then using that to combine across your multiple recordings. So what this allows us to do then is to average our data across all of these recordings, even though our probe is in a slightly different location every time. And then we can make some statements about the dorsomedial, dorsicentral, and dorsolateral striatum. So for example, if we were to look at task responses, we could see we get nice visual responses in the dorsomedial striatum. If we look at movement responses, we get nice movement responses in the dorsicentral. And then we get nice licking responses in the dorsolateral striatum. The other thing that it allows us to do is ask how good a job we are predicting striatal activity from cortical activity. So in other words, I came into this project expecting that there would be some kind of transformation from the cortex to the striatum, or the striatum might be doing some kind of unique stuff. But what it turns out is that if you are able to record both striatal and cortical activity, you can figure out what the striatum is doing just by knowing what the cortex is doing. So in this case, I can take all of the trials where I record from these different segments of the striatum, and I can look at their striatal spiking. In this case, the mice are doing an IBL-like task, which is what you just heard about from Anne. If I try to predict this activity just from the cortical activity, I can do a pretty great job. In fact, I can do just as good a job predicting the striatal activity from the cortex as I can from the task, for example. So this allowed us to answer this scientific question of, is there a nice relationship from the cortex to the striatum? Is there some kind of transformation? Are these things doing different jobs? It turns out that there is a really nice relationship, and it's defined by topography, and it's essentially just this simple one-to-one relationship. So doing these two modalities at the same time allowed us to answer this nice scientific question, in addition to being able to do things like align exactly where your probe was on a given recording. I'll just quickly mention that one of the nice things it also was able to show us is that there's a change across learning. So if we look at trained animals versus untrained animals, the visual cortex doesn't show a difference in activity when you show a visual stimulus, but the dorsomedial striatum has a much increased response to the stimulus after learning. So in other words, you always have a nice topographic relationship from the cortex to the striatum, but it looks like the strength of the propagation of activity increases across learning, so that the visual cortex starts to drive the striatum more strongly after learning. All right, so that's the end of that. The ideas that I want you to walk away with are that even if you're not doing imaging in your lab, one of the things to keep in mind is that as kind of Anne was talking about, every time you put your probe in, you're going to put your probe in a slightly different place. So it's worth thinking about how are you going to group your activity across recordings. If you're in a big diverse structure like the striatum, then it's important, as opposed to if you're in a smaller structure, or you're able to do something like retinopathy if you're in the visual thalamic nucleus or something. So it's important to think about where you are for each recording and how to group those. One of the ways to get at that is by combining imaging and electrophysiology. The other thing is that if you're planning on doing any kind of light related things, you're going to get these big artifacts on the probe. If you ramp the light up and down rather than just switching it on and off, you're able to get rid of a lot of this artifact. And if you do common average subtraction, you're also able to get rid of enough of the artifact that you can just cleanly sort these things out in Kilasort. So after me, Maxime is going to give a talk about optogenetics, where he'll have to face kind of similar things, but for a slightly different purpose. So probably a lot of you will use optogenetics. In that case, these principles are things that you'll be able to apply in your experiments. Okay, so I can take
a few questions. Do you mind answering those questions in the Q&A?
Sure. So I'll just run down these. So Joe asks, are the light artifacts depth channel dependent? Whoops, I'm going to say answer live. Yeah, so they are to an extent, although not in a way I think that makes a whole lot of sense to me. So I thought, for example, that if you have part of your probe out of the brain, part of your probe in the brain, the part out gets a lot more light, and so it's going to have a bigger light artifact. Turns out that's not actually true. So I don't know if the light artifact kind of like travels the length of the probe or something. I stay I keep that ambiguous. And instead, the way that I deal with each channel and each light color is by doing this local average referencing. So if you just take the nearby 50 channels and you subtract the average, it does a pretty good job. That case, it also doesn't matter what the color is because the blue and the violet in my case are going to be alternating, but it's going to be the same across all channels. So just doing something really simple like this local referencing is good enough to get rid of those things. All right, Harsh asks, do you have a repository with code for this processing? I do. I think probably the easiest way to get at that is by the Slack channel or by email. It's kind of niche code, so it's not super clean at the moment, but feel free to email me specifically and I can provide that for you. All right, Lewis says, what is the sensitivity of the Neuropixels to infrared light? Good question. I don't know if Nick or Flora, if you guys use, so we have infrared lights that we use to illuminate the mouse when we're doing our video recordings of mouse behavior. The light is on all the time, and if the light is on all the time, then nothing happens. So I'm not sure if there's sensitivity to that if it's flickering.
I think there definitely is some sensitivity, but I definitely also don't know the relative sensitivity of infrared to some other wavelengths.
Yeah. Joe asks another one. Since we know the biophysical properties of the GCamp and have info on their decay, how is using the ephys trace different for deconvolution than using the temporal properties? It's not, it's just empirical. So I think if you knew exactly what the GCamp was doing and you tried to turn that into a deconvolution, hopefully you'd get exactly the deconvolution kernel I came up with. So I was just trying to be agnostic as to what it was and use the best empirical thing. So hopefully it's the same kind of thing. Andwan asks, for this experiment, does it make more sense to use a wide field setup on a Neuropixels recording rig or add Neuropixel setup onto a wide field rig? Yeah, chicken and egg. I think we kind of just did it both at the same time. I mean, the Neuropixels setup is small, right? It's a computer and this little box, and then up at the experiment section, you've just got a pole and your manipulator. So I think moving the Neuropixels stuff kind of around is doable. But in my case, I set up both of them at the same time. All right, I think we got time for a couple more. Which version of GCamp are we looking at? This was all 6S. This mouse that we're using was developed by Joe Wexelblatt, who has been the one asking half the questions. It's this totally amazing mouse that expresses GCamp 6S under the TETO promoter, the CamK2 promoter using a TET system, and it's totally awesome. One of the things that you might be aware of from our lab is that if you have a pan-neuronal expressing GCamp6F, then you tend to get epilepsy. So we stay away from those mice. We don't use 6F, we use 6S. All right, Ensor asks, if I do common average referencing, should I disable average subtraction on Kilasort? I didn't know there was a thing on Kilasort to do that. I do them in separate stages. So I always subtract the average first, and then I run it through Kilasort. If Kilasort does the same thing where it subtracts the average across all channels, then it's probably redundant. So I guess you might as well keep it on if it's not going to make anything worse. That's what I would suggest. All right, Mingxi asks, may I ask whether there is any common function that allows us to do local channel median subtraction? So Nick came up with one that I think I might still use. The function is kind of simple. It just opens up the DAT file and then subtracts the median of everything or locally. So if you email me, I can provide that to you. I'm not sure how clean it is for general use, but it should be okay. All right, and the last one, Jaspreet asks, are there other imaging techniques apart from 2-photon or wide field have you tested along with the Neuropixels? Would it be possible to try with a Gren lens? Yeah, so no one in our lab has tried that. I think you'd probably run into the same challenges because whenever the light hits the probe in any case, you're going to get this thing. So we haven't tested it, but I assume it would come with pretty much the same kind of challenges. All right, and that's my time exactly.